Okay, good morning. Today we are starting the very new chapter, that is heredity and evolution. Now in this chapter we will going to talk about the characters and the process of their transmission from one generation to another generation. That processes and what are those characters which are transferable from their parents to their offspring, and what type of dissimilarity and how these dissimilarities are appearing among the member of a particular species. So this is all about we will going to discuss in this chapter. For example, a particular organism or particular member of a population in a species population is showing the dissimilarity with their another member. So this is known as variation. Then the question arises why these variations are coming or how these variations are transferring from generation to generation. That means the things are also visible in our environment, in our surrounding, that we all are not similar to our parents, even we are not similar to our siblings. So why these dissimilarities are happening or appearing in the member of same population or same species population. So in this chapter we will going to discuss about the reason behind all this and the mechanism how these variation or how the characters are actually transferring from one generation to another generation. So now the most important term, that means the chapter name is heredity and evolution. First we will going to discuss about the topic heredity in this chapter, and then we will continue with the evolution. Now what basically means is heredity? Heredity means the characters that are transferred from one generation to another generation. So here the heredity are the characters that are transferred from one generation to other generation. Fine. That means these are the characters which are transferable. That means the character that actually transferred to the next generation is known as heredity. But there are certain process or mechanism or the method through which the characters are transferring from one generation to other generation. That is known as inheritance. So inheritance is the process of transfer of character from one generation to other generation. So this is the process of transmission of character from one generation to other generation is actually known as inheritance. Now the term arises variations. What is all about the variation is? Variation means the dissimilarity, the changes. That means dissimilarity or the changes which appearing in the members of a particular species population. So these are the dissimilarities among members of a particular species population. Fine. So these are the variation. Now there is a particular branch of biology known as genetics. This is a branch of biology that deals with the study of inheritance and variation. That means the branch of biology that deals with study of inheritance and variations. That branch of biology under which or inside which we are studying about these processes, that is how the inheritance is happening and how these variations are appearing. So that branch of biology is known as genetics, and in this chapter we will going to discuss about the genetics only. Now let's discuss firstly about the variations. Basically these variations are very important because the variations when comes, then only the species evolution has been arised. That means you can say variation when enters into a particular species population, then only different characters will going to reveal in generation to generation. Now basically what are these variations all about and how many types of variations are there? Variations means changes. That means we all know a particular species always having different members will always be, does not mean always that they are similar. They may show some dissimilarities also, and the dissimilarity may appear in terms of different parameters, maybe in terms of their color, maybe in terms of their height, maybe in terms of their genetic makeup. So there are certain characters which may be different in different member of different species. Like we human beings are very much dissimilar to each other, although we belongs to same species, but we are not all are same, we are dissimilar. That means there are lot of variations inculcated in our human population. So these variations, actually some variations are transferable and some are non-transferable from one generation to another generation. Then on that basis also we have classified the variation into two types. Variations that are dissimilarities, which may or may not be transferable. So on that basis we have classified the variation into two basic types: the somatic variations and germinal variations. Fine. Now what is all about this somatic and what is this germinal? As we all know, in all the organisms, whether it is plant or we animal or we human being, we have two types of cells. Two types of cells. That cells are one somatic cell and the germ cell. Now what is somatic cell and what is this germ cell? The basic difference between the two type of cell is this somatic cell having the chromosome number or the chromosome set as diploid, and the germ cell have the chromosomal set as haploid. Now what does it mean is, our whole body or any organism having all its cell are all somatic cell. That means the cells which are involved only in the growth and the development and also in the repairing in case if tissues get damaged, like our skin cell, like our organ cell, our eyes cell, our lungs cell, our respiratory system. That means we have all different type of system. The cells are all of these system made up of somatic cell except the reproductive system. Generally in the reproductive system we have different reproductive organ. Animals do also have different reproductive organs. That is, the male reproductive organ is different and the female reproductive organ is different. Same is in the case of plants also. So in that case, the cells which form or which develop by the process of cell division known as meiosis, and then after meiosis, when the cells produced with their haploid set of chromosome number, then those cells are known as gametes or gametic cell and is also known as germ cell. So the somatic cells does not involve in the process of fertilization, but the germ cell which may be or which may develop in the form of gametes. The male gamete is different and the female gamete is different. These gametes when fused together later on to form a diploid zygote, then only the generation will transfer or the character will transfer. Fine. That means the germ cells involved in the process of fertilization, while the somatic cell does not involve in the process of fertilization, but they involve in the process of growth and development. Now the somatic cells and the germ cell, these two type of cell, they decides whether the variations are transferable or not. Now these variations that are somatic variations, they are non-transferable. That means here if variations done, then these are not transferable. That means these type of variation, if any changes in the genetic material, that is in the DNA, like DNA is the most important genetic material inside our cell. Like you people have studied about the chromosome, about DNA in your last class, that is 9th class, in a very first chapter, that is cell. So you have studied about the structure of nucleus as well as all the content of the nucleus, and since the nucleus contains our genetic material in the form of chromosome, like again if I'll revise it, so this is our nucleus and it contains all the genetic material in the form of DNA. These are all DNA threads. But these DNA threads actually they combined or again coiled up to form a structure known as chromosome. So this chromosome is made up of DNA only, and the segment of the DNA is known as gene. Fine. That means you can say inside the cell we have nucleus. The nucleus is the controlling center of our cell. Nucleus is the headquarter of the cell which controls the activity of cytoplasm. But this nucleus also contains our genetic material, that is DNA. This DNA coiled or these chromatin thread, DNA is also known as chromatin thread, along with the protein they are known as chromatin thread. These chromatin threads when condensed or attain or appear in a form of particular shape, then it is known as chromosome. And this chromosome is actually made up of DNA thread, and the segment of that DNA is known as gene. Fine. Why it is known as gene? This gene actually contains an information for a particular character, particular product or a particular protein. So this gene is having the information or the storage of information of formation of a particular product or a character to express. So here what we are talking about is that if the variations, that means if there occurs changes in the gene part of DNA present in the nucleus of somatic cells, that means अगर कोई change उस DNA के gene में हो रहा है जो कि उस somatic cell के nucleus में present है, then these variations or these changes cannot transfer from parents to their offspring. Are you getting it? Basically what I am trying to say is कि अगर somatic cells के gene में कोई भी तरह का change होता है, variation होता है, due to any factor, due to any reason, then these changes cannot transfer from parents to their offspring. Why? Because somatic cells of one parent does not fusing with the somatic cell of another parent to form a zygote. That is why the somatic variations are not transferable from one generation to other generation. Fine. So these variation, if done in the somatic cell then these variation, these changes are not transferable from parents to their offspring, and this is known as somatic variation. But if variations done in germ cell, like here in somatic cell, in germ or gametic cell, then the variations transfer from one generation to other generation. Fine. So, in case of germinal variation, the changes in the gene part of DNA which are present in the gametic cell or germ cell, then that changes can be transferred from parents to their offspring. Why? Because these gametic cell and the germ cell will going to fuse during the process of fertilization to form a diploid zygote. So that diploid zygote will definitely contains the information of both types of gametic cell. That is why these variations can be transferable from parents to their offspring. But it was not happening in case of somatic cells because they were not fusing together. So this is the basic difference between the two variations, is that somatic variations are not transferable while germinal variations are transferable. Now let's talk about the somatic variation, the reason behind the somatic variation. Basically, the somatic variations factors. There are certain reasons behind the somatic variations, factors affecting it. Fine. That means somatic variations is the variation which actually appear in an individual during its life and it get lost with its death. That means जब तक वो जिंदा है वो ऑर्गेनिज्म, उसकी पूरी लाइफ के दौरान उसने जो भी वेरिएशन या जो भी चेंजेस आए हैं वो उसके उस लाइफ तक ही सीमित है, उसकी उस लाइफ तक ही लिमिटेड है. इट इज़ नॉट ट्रांसफरेबल टू द नेक्स्ट जनरेशन. फाइन. दैट मीन्स वो चेंजेस, वो वेरिएशन उसके अंदर किसी न किसी कारण से आए हैं. सर्टेन फैक्टर्स which are the reason or which may be the reason for causing those variations. So those factors which are responsible to cause those variations in case of somatic variation is, it may be environment, mean the environmental factor. Now environmental factor, basically these environmental factor includes light. Here like I am writing, it includes light. It may include the habitat. It may include nutrition. Fine. And there are lot more other factor. Fine. So environment means the area which formed by two component, biotic and abiotic component. Environment is our surroundings which formed by two component, that is biotic and abiotic component. Now as we all know, biotic is living and abiotic is the non-living component. Here we are talking about abiotic factor because we all are biotic component on which the effect of these abiotic factor causes these type of somatic variations. Now here if we talk about light, so light is the very good factor which definitely causes the variation. For example, if I say like the person or a particular organism or species, if they are living in or under the sunlight, fine, like we are also, in case of we human being also, our body when exposed to sunlight or more exposure is done to the sunlight, then the melanin pigment present in our skin cell gets more activated and this melanin pigment will going to be spread during the presence of sunlight and it causes the darkness of the skin. But on the other way, this melanin pigment also helping us to protect from the ultraviolet rays of the sun. So due to light, what is happening? There is a change is taking place in the DNA part because the melanin pigment is a protein pigment actually, and the proteins information stored in gene part of DNA. So due to influence of these light, this light actually affects the expression of that gene which carries the information of synthesis of melanin pigment. So what is happening here? This light is affecting the gene for melanin pigment to express. Fine. And the role of melanin pigment is, we all know that it stimulates the darkness of the skin. That means whenever the melanin pigment expresses, it spread all over the skin, and due to its spreading, our skin gets darker. But the other important role of this melanin pigment is to protect our skin from UV rays, because this melanin pigment is made up of, this melanin pigment actually cover our underlying epithelial cells from the action of UV rays by the sun. Fine. So in this manner, the variations is there, that in few people, this melanin pigment, in few organisms, the melanin pigment spread more, and in few, the melanin pigment does not spread more. But that variation is not transferable. This secretion of melanin pigment, which is being increased only due to presence of more or due to more availability of light with which our skin cell get exposed. So that variation is not transferable, because it is present in our somatic cell. The gene which is affected present in our skin cell, and the skin is a somatic organ. In the same way, habitat is also affecting the genes of somatic cell. For example, whatever in the habitat, like in habitat, for example, some organism during evolution, when the life was originating on the earth, and when there was lot of variation, when there was lot of changes was occurring over the earth in different organism, in different species, at that time also, when the species were changing their habitat, that means few were living in the water, and when they comes on land, they have changed their mode of living, mode of style. So obviously, but that changes are not transferable from generation to generation. Fine. So that habitat is also affecting the variation, or affecting the, or causing the somatic variation, which is not transferable. In the similar way, the nutrition, that means the how the food, process of obtaining food. This nutrition means process of obtaining food. Fine. The process of obtaining food and utilize it to obtain energy in the form of ATP, that is known as actually nutrition. This is also not transferable. Due to nutrition also, when any variation causes in a particular gene, that means when if any organism is feeding on a particular type of food substance, and if it's changing its food habit due to availability of food, that means if it's more important food which is not available to them, then they will going to feed on another type of food. Like in food web happens, the animals, if they are not getting their liking food, then they will going to feed on another food. Like in case of dog, for example, in the dog, basically they started eating, like in case of bone, if they are not available to them, then they will start to eat the chapati or something veg food also. So in that case, the variations if comes in their cell, in their cellular part, like if the variation come in their teeth, like structures of teeth also, then that is not transferable from one generation to another generation. So feeding habit if changes in any species, or the mode of nutrition if changes, because the mode of nutrition take place in the digestive system, so digestive system is again the somatic cell, so that variation is again not transferable. Fine. Now this is about environment. This is one of the important factor which actually causes the somatic variation. Another factor may be use and disuse of organs. That is, if we are using a particular organ more, that means we are talking about the somatic organ over here. If you use a particular somatic organ more, then it is not transferable. Like, and if we are not using a particular organ a lot, then the characters are not transferable. Like in case of long neck of giraffe, like according to a particular scientist Lamarck, he said that the giraffe was using its neck more, or the organ neck more as compared to other organ, because it was continuously stretching its neck. It was continuously stretching the neck to feed on trees, that means to feed on the heighted plant, heighted trees. That's why he said that the organs character is being transferable, but it was not happened as such, Basically, if you are using a particular organ and if you are not using that particular organ, then the characters are not transferable from one generation to another generation. So that is also the factor. That means if particular organ you are using a more, then that character is limited to that generation only. It is not transferable to the next generation. Third important factor is basically the conscious efforts. These are the efforts done by we human beings. These are the artificial. That means the efforts done by man-made. Like we human being, what we are doing nowadays, we are causing certain changes, genetic changes we are causing in certain plants for getting more and more food, or for getting more or high yield of food in order to supply or in order to satisfy the demand of food. So these are the efforts again done by we human being, artificially done, artificially we are doing on the plant, but that is again not transferable to the next generation because that we are doing in their somatic part only. So these are the certain factors which are causing in case of somatic variation. Germinal variation we have also discussed it. Now we will going to start that what are actually the significance of variation, why these variations are very, very important. The most important significance of variation is basically due to these variations, due to the variation, evolution of species was done. Generally, when the earth was originated and when the life was originated on the earth, at that time the different species were there, but due to changes in the environmental condition, various changes are transferring from generation to generation, and the species which has been arised or which has been evolved, it entirely different from the ancestral species. So this is very important also to bring about the evolution. Secondly, due to variation only, the lifespan or you can say species become more stable. That means you can say the surviving period or the survival of the species has been increased. Why? Because if in a particular species, if the variation does not occur in them due to fertilization or due to any reason, if there is no variation in them, then such a species cannot cope up with the changing environmental condition, because environmental condition always keeps on changing. But if variations are happening in such species population in different members in their member, then they will be going to adjust with the changing environmental condition also. So the survival period of those species in which the variations are present or variations are appearing, they are more surviving as compared to the species which are in which the variations are not going on. Fine. So basically the evolution of the species and species becomes more stable, having more survival period is the basic or most important significance of the variations. As well as due to the variations, most important or you can say new traits appear in generation. Again, some new character may also appear due to variations in the generation, and these new trait again that support the population, that these new character may support the species population again to adjust with the changing environmental condition. So basic things is the most important significance is all about evolution and the species survival and stability attained due to the variation. Now we are going to discuss about the genetics. Basically here we will going to talk about, before proceeding with the genetic, our in our mind one name of scientist comes, basically that is Gregor John Mendel. Gregor John Mendel was a father. He is a father of genetics. Actually, he is considered as a father of genetics because he was the only one who introduced genetics by working on the garden pea plant, Pisum sativum. Fine. Now, before proceeding to his processes or how he proceeds or how he experimented on the pea plant to explain his principle and to explain the genetics part. So, first of all, we should understand about certain different terms related to it. That means the terminology or terms which Mendel has also used to explain his principle, to explain his theory by working on pea plant. So, let us discuss about first of all the terminology related to genetics, then later on we are going to proceed with the processes or principles of the Mendel or the life history of Mendel. So, now we are discussing the terminology. In terminology, the most important term, first of all, first term arises gene or factor. Now, when Mendel was studying or when Mendel started working on this field, on working on the pea plant, then he did not use or did not know about the term gene, he did not know about the term DNA or chromosome, because up till that time microscope was not actually discovered, because at that time the Mendel was not able to observe anything inside the cell under the microscope. So, he used certain different terms. He considered there is certain something or certain factor present inside the cell in which the information of a character is located, and he used the term factor, not gene. The term gene later on used by another scientist. So, factor means anything that carries the information of a character present in the cell is known as factor. Fine. This term factor used by Mendel. Second terminology he uses allele. Now, what is allele? Actually, Mendel told about that every character have their alternative form. That means every character have their you can say the alternative. That means if a particular character is height, then it has two types of height, that is whether the organism is tall or whether that organism is dwarf. So, here this allele he introduced or he explained about this, that this is the alternative form or alternative pair of a particular gene that is located on the same locus of homologous pair of chromosomes. What he said is that this allele is defined as alternative form or factor of a gene located on same locus of homologous pair of chromosome. Fine. That means he said ki for example height is a character, then it has two alternative form, tall and dwarf. Now this tall and dwarf is actually like we have chromosome in their paired form, because the organism have diploid cell and the diploid cell have chromosomal pair form, and these paired form of chromosome is known as homologous pair of chromosome because they are similar in structure, shape and size. Now this chromosome have the gene part, and this gene part is having the information of this particular character height, but this character height either present in the form of tallness or they may be present in the form of dwarfness. So this tall trait, the tall variety obviously appear in that paired form, and dwarf variety also appeared in their paired form in the diploid set of cell. Basically in a diploid cell, every variety obviously appear on chromosome in the form of pair. So this pair form of this variety, that is TT, like this tall represented by T and dwarf represented by small t, like symbol we are using here. So this capital T if presented on the same position of this homologous pair of chromosome, then this is known as one pair of allele, and one T is known as one allele and this allele is known as pair of allele. Fine, and this pair of allele present on the same locus of homologous chromosome is appearing as a tallness or a tall variety of that particular organism. Fine. Similarly, if dwarf, then it's small t and small t. So this is the location actually where the information of a particular variety is located is known as allele or a pair of allele. Third terminology is dominant trait and fourth is recessive trait. Now what is this? Mendel has said that if any trait means variety of a particular character, like height is a character, then it has two traits, tall and dwarf. Fine. So if the trait of a particular character, if it appearing in each and every generation with higher frequency, then that is known as dominant trait. That means here the trait that appear in each and every generation with major population of a species is known as dominant trait. But if the trait that appears in like that do not appear in each and every generation and can skip the generation, and if they are appearing, they are not more than that the dominant trait, that means the population found to be lesser in a particular generation, then that trait is known as recessive trait. So the trait that can skip the generations and do not express in their in their heterozygous condition. Such trait is known as recessive trait. That means the traits that can skip the generation and they are not appearing in each and every generation and cannot express in their heterozygous. That means the recessive trait are suppressed by the presence of the dominant trait. Like tall is a trait and dwarf is another trait. But these are the trait of character height. Fine. Now height is a character. This is a character. But these are traits of this character height. Now tall and dwarf. Tall can express by two methods. Either by capital T or by capital T small t. But whenever this capital T comes, because the tall represented by capital T and dwarf represented by small t, it always appears be tall because this is the dominant trait. Tall is a dominant trait, and whenever it comes over the DNA or appears on the gene part, then always the trait appear or express is tall only. But dwarf will express only and only when it comes or express in the homozygous condition. That means whenever both the allele on that gene locus of homologous pair of chromosomes, if both the allele appear on them is of recessive trait only, then only that recessive trait is going to express, otherwise the recessive trait will not going to express. So that is why the trait that can skip generation or may appear only if they comes in their own homozygous condition. This is known as recessive trait. Now the next terminology is homozygous condition and heterozygous condition. Fine. What is this homozygous and heterozygous again? This homozygous condition means homozygous means the zygote form by fusion of two gametes with same type of allele. That means here, whenever the zygote is forming, that zygote when formed by a fusion of two such gametes having similar type of allele, whether it contains both the allele dominant or both the gametes either contain recessive allele, then the zygote form is known as homozygote. So this is like if this gamete is having only capital T allele and this gamete is also having capital T, so zygote formed is always diploid cell. Whenever it forms due to fertilization or due to fusion of these two gametes, then this zygote is known as homozygote, or it may appear in this form also. So this is also homozygote because the same type of allele are fusing together to form a diploid zygote. So thus such type of zygote is known as homozygote. But heterozygote may just that things becomes different. Here the zygote forms by fusion of gametes with different type of alleles. That means one gamete is carrying dominant allele and the other gamete is carrying the recessive allele. Then only the zygote when formed is known as heterozygote. So this is heterozygous condition and the zygote formed is heterozygote. Fine. So this is homozygous condition, heterozygous condition. So this is all about the basic crosses or basic terminology. So this is all about the basic terminology regarding genetics term. Now the few of the terminology are also given, but that we will going to discuss in our next lecture when I am going to explain you about the monohybrid, dihybrid crosses. Along with those crosses also we will going to discuss about the back cross, test cross. Okay, thank you.